And thank you, Callie, for volunteering to present. And she's going to be talking about the novel role for the calcineurin phosphatase at the nuclear pore complex. So with that, you can go ahead.
Awesome. Let me, sorry.
Oh, and Jesse, record. Okay, good.
I did.
All right. I am successfully screen sharing, yes?
Yes.
Okay, perfect. All right. Well, thank you guys so much for being here today. I'm really excited to share the story that actually part of which was recently published in Molecular Cell. I've got a nice little plug here on my title page. If you guys are interested in looking into this story a little bit more deeply, you can go check out our paper. It was published this week and we're really excited about it. So today what I'm going to be doing is giving you kind of a quick overview of how we approached this project of really systematically mapping the calcineurin signaling network in human cells, and then getting to the point really quickly that we found this novel role for calcineurin at the nuclear pore, which I'm going to spend the majority of my time talking about today. And I want to start out by introducing calcineurin, which is the calcium and calmodulin activated serine threonine protein phosphatase, which is best characterized as the phosphatase responsible for dephosphorylation of the NFAT family of transcription factors and subsequent immune cell activation. And so we know that calcineurin plays a really critical role in the immune system. And actually, the widely prescribed immunosuppressant drugs, FK506 and cyclosporin A, target calcineurin specifically to prevent the rejection of solid organ transplants. However, we also know that calcineurin is ubiquitously expressed and plays really important roles in other tissues like the brain and muscle. And so therefore, it's not so surprising that when you have inhibition of calcineurin in all tissues during immunosuppressant treatment, which is typically lifelong, you additionally get many adverse side effects of immunosuppressant therapies, a few of which I have listed here. So these side effects are likely due to inhibition of calcineurin in non-immune tissues. And so we hypothesize that identifying and mapping the spectrum of calcineurin substrates and regulators in human cells would not only allow us to map the human calcineurin signaling network, which is just a really interesting basic cell biology question, but also to maybe begin to elucidate the underlying molecular bases of these diverse side effects that patients experience when on immunosuppressant drugs. Before I jump into our approach of how we're going to solve this problem, I wanted to tell you a little bit more about calcineurin and its structure and how it recognizes substrates. So this is a crystal structure of calcineurin, which is a heterodimeric enzyme with a catalytic A subunit shown here in gray and the catalytic site highlighted here in light blue, and then a regulatory B subunit, which is shown here in kind of a tannish gold color. So then here is that same crystal structure. So you can see the catalytic and regulatory subunits here. But now this is co-crystallized with the viral inhibitor protein A238L. So, and that's shown here in this purple ribbon. What's also highlighted here in yellow and in green are the two conserved surfaces on calcineurin that bind to the two short linear motifs or SLIMs termed PxIxIT and LXVP, which are named such based on the sequence composition of known sites and calcineurin substrates. And that's what I'm showing you here are logos of these SLIMs with the core PxIxIT and LXVP residues, as well as some flanking residues N and C terminal that we found also are important for substrate recognition. So as I'm sure many of you are aware of, working on phosphatases, SLIMs are a type of sequence motif that are defined as being degenerate sequences that are found primarily in disordered regions of the proteome. They mediate many protein-protein interactions, especially in phosphatase substrate signaling networks, I'm sure you guys are familiar with, and they are rapidly evolving. So because we know that calcineurin interacts with substrates via these two PxIxIT and LXVP motifs, the way that we're approaching this problem is to go to the proteome experimentally and computationally and identify proteins that contain calcineurin binding SLIMs using various approaches. So to address this goal, we're taking a SLIM-directed, multi-pronged approach, which is kind of represented here. So the goal is that each of these individual approaches, which I'll walk through in the next couple slides,
talking about
that each of these approaches individually would identify potential calcineurin substrates via their SLIMs, but that they would also synergize with one another to allow us to identify the human calcineurin signaling network, which I will occasionally refer to affectionately as the calcineurin Nome, because in molecular biology, everything needs an ohm these days. I also want to take a moment to point out here that this approach, while we applied this to calcineurin signaling, it really sets a precedent for any SLIM-based interaction network. So this approach or something like it could be used for other enzymes and proteins. Something to think about with your projects. All right, so the first of these approaches is proteomic peptide phage display, which utilizes a phage library containing all disordered regions of the human proteome. These phages are incubated and enriched against purified calcineurin, which we're schematizing here as being shown on the bottom of a dish. So you incubate these phages against purified calcineurin to directly select for phages that are displaying calcineurin binding SLIMs. So upon sequencing these phages, we identified dozens of PxIxITs and LXVPs, some of which were previously known, which is really good, but many that are novel. And a fellow researcher in the lab, Jaggery Roy, who's the co-first author on this paper with me, she validated each and every one of these novel PxIxIT and LXVPs through individual in vitro binding assays. So what's really exciting here is not only that we identified so many novel PxIxITs and LXVPs, but that each of these peptides really represents an individual protein that could be regulated by calcineurin. And so we've not only identified candidate substrates, but more specifically the precise motif that calcineurin might be using to interact with them, which is really powerful when you're performing downstream functional analyses. So just to give you a visual representation, before we started this study, our collective understanding of calcineurin binding SLIMs is represented here. And after just the ProPD analyses, we've more than tripled our understanding of calcineurin binding SLIMs. As I mentioned just a minute ago, each of these peptides could represent an individual protein that's regulated by calcineurin. For example, on this list of PxIxIT peptides that we identified is the nucleoporin Nup153, which is a key transport nucleoporin and something that I'm going to talk a lot more later in the talk. In a little vignette that I don't really have time to talk about today, we also identified an LXVP from the key developmental transcription factor Notch1 and went on to show through a series of experiments that that LXVP is functional and that Notch1 is a novel substrate of calcineurin. So if you're interested in learning more about that, you can go check out our paper. Ultimately though, what's really great about the ProPD approach is that it identified or tripled our number of calcineurin binding SLIMs, which allows us to build a very sophisticated computational algorithm to predict more novel PxIxITs and LXVPs from the proteome. And so to do that, we used this wealth of understanding that we just gained of these motifs to generate two different position-specific scoring matrices, or PSSMs, that are used to map and score peptides throughout the proteome and generate a list of sequences that score highly against our PSSMs. So from there, we could go on and filter for criteria like accessibility to calcineurin, because we know where calcineurin hangs out in the cell, the disorder score, because we know that disorder is a hallmark of SLIMs. So using this approach, we've identified hundreds of proteins that contain at least one high confidence LXVP and or PxIxIT. And then to validate this approach, we performed five-fold cross-validation of these PSSMs back against the proteome and observed highly significant performance of these motifs, which validates the approach and supports our confidence in these computationally predicted calcineurin binding SLIMs. And then finally, another way to identify calcineurin substrates, but also validate our PSSM approach, is to generate an independent experimental data set of calcineurin interacting proteins. And in our case, we use proximity labeling, or BioID, which is a really powerful tool to identify low affinity or transient protein-protein interactions in vivo. So to do this, and what I've got a little cartoon of up at the top, we fused calcineurin to the promiscuous biotin ligase BRA, and expressed it in HEK293 cells. We also took a really neat approach because, again, we know the conserved docking surfaces on calcineurin where it's binding to PxIxIT and LXVPs. And we created BRA calcineurin fusions that are defective for PxIxIT binding or LXVP binding. And eventually we'll be able to look at any differences in biotinylation between a wild type and what we would call a docking mutant. So once you express these BRA fusions in hex cells, label them with biotin, and then ultimately do a biotin enrichment and mass spec analysis, we identified almost 400 proteins that were biotinylated above background with the wild type calcineurin fusion protein, including, very importantly, many known calcineurin substrates, and also about 50 proteins that were identified in our PSSM analyses, which is a really significant enrichment and supports our confidence in both the BioID approach as well as the PSSM. Furthermore, we also identified a large number of proteins with reduced biotinylation with the docking site mutant data sets, which is what I'm representing here in this graph. You're looking at the log2 ratio of biotinylation between the wild type calcineurin fusion and either the PxIxIT docking mutant or the LXVP docking mutant. So if you expect there to be reduced biotinylation with either of the docking mutants, you would anticipate those proteins partitioning to this upper right quadrant of the graph. And that is where we see a large number of significantly biotinylated proteins, including a lot of known calcineurin substrates and interactors, which I'm highlighting here. And then the colorful circles are showing you the proteins that performed or that we found SLIMs via our PSSM approaches. So you can see that those are really nicely partitioning along with that docking site dependent biotinylation. In another little vignette that I don't have time to share today, through our BioID analyses, we also identified a large number of highly biotinylated PxIxIT dependent proteins at the centrosome, a site where calcineurin localization or function has never been attributed. So we have a graduate student in the lab who's currently working to understand the potential role for calcineurin at this important structure. So altogether, coupling the ProPD, PSSM, and BioID analyses with a list of previously characterized calcineurin substrates and interactors, we have built the first comprehensive signaling network for a phosphatase in human cells, and we're calling this group of proteins the calcineurinome. So what I'm showing you here are the Go terms that are significantly enriched within the calcineurinome. We see enrichment for processes in which we know, we already knew that calcineurin plays a role, like ion transport and transcription. So not only do we identify many novel candidate substrates within those terms, kind of expanding our understanding of what calcineurin is already doing there, we also identified completely novel areas of calcineurin biology, like nuclear transport, as I'm going to tell you about in the next few slides. So when we first identified nuclear transport, we were a little bit surprised because no one had ever, identified a role for calcineurin in this. However, when we started digging through the literature, it became pretty clear that the nuclear pore is an area that's kind of ripe for regulation by calcineurin. For instance, we know that nucleoporins are really heavily phosphorylated and that phosphorylation regulates transport through the pore. We also know that nucleoporins are heavily phosphorylated during mitosis, and that regulates nuclear envelope disassembly. Additionally, changes in calcium levels can affect the overall structure of certain nucleoporins, one of which I'll tell you a little bit more in a few minutes. So this seemed like a really exciting novel area for us to follow up on. I want to just give you a quick introduction to the nuclear pore complex, which is shown in cartoon form here. You're kind of looking at a lateral view if you had cut through the nuclear envelope, looking at it from the side. The nuclear pore is a really large proteinaceous structure that straddles the nuclear envelope and provides a conduit for proteins and RNA to travel between the nucleus and the cytoplasm. Each individual pore, of which there are thousands on a mammalian nucleus, has more than 30 individual nucleoporins that make it up, and they're typically present in an 8-fold rotational symmetry, which comprises that ring-like structure that you see if you look straight down the nuclear pore. So shown here in bold, as well as in color down below, are proteins that we identified from our computational and experimental analyses as having calcineurin binding slim. And what was interesting when we first saw this was that the majority of these nucleoporins and transport factors that we identified are proteins that are directly involved in nuclear transport, suggesting a potential role for calcineurin in this process. I also want to point out here that while our paper was in review, another really nice paper came out that identified several interactions between calcineurin and various nucleoporins, really supporting this idea that clearly calcineurin is present at the pore and is performing some function, which was really exciting for us. So again, this suggests that there's a potential role for calcineurin at the pore. And just as a jumping off point, we wanted to see whether any inhibition of calcineurin could affect the rate or extent of transport of a commonly used nuclear transport reporter, just to get a feel for whether calcineurin was actually affecting transport. And we decided to use the nuclear transport reporter that's called RGG, which is a glucocorticoid receptor GFP fusion protein. And it's unique because only upon treatment with dexamethasone, which is going to bind to the glucocorticoid receptor region, only upon treatment with dex does it localize to the nucleus, as shown here on the right. So you can really get, you can gain insight into import rates and then extent of accumulation over a time course. So we can perform live cell imaging on cells expressing this reporter. And these cells would either be treated with a vehicle control or with calcineurin inhibitors, and then look to see if there are any differences. And that's exactly what we've done here in these videos. So you've got Hela cells. We're showing 2 cells from each condition, the DMSO, FK506, or cyclosporin. And these are Hela cells that are imaged every 30 seconds over 90 minutes. And what you can see is that the extent of accumulation of this RGG reporter between the nucleus and the cytoplasm is much higher in the cells treated with calcineurin inhibitors, suggesting that calcineurin typically positively regulates nuclear import, and we've interrupted that by inhibiting calcineurin. I also want to show these graphs, which are just quantifying these videos over many hundreds of cells and showing that both of these inhibitors behave really similarly in slowing down the or lowering the extent of nuclear to cytoplasmic distribution, but also significantly impacting that initial rate of accumulation. So this is a really exciting, preliminary result. It got us really fired up thinking, okay, calcineurin clearly is playing a role here, what's going on, but it doesn't tell us anything about mechanistically what's happening. Is calcineurin impacting nucleoporins? Is it impacting transport factors? Are certain proteins differentially phosphorylated? All of these open questions still existed. So to begin to address this, we kind of went back to what we had learned from our calcineurinome analyses and observed that all 3 components of this nuclear basket structure, shown here in these colorful cartoons, each contained a high confidence PxIxIT motif, suggesting that calcineurin likely interacts with them. We have confirmed that these sequences mediate PxIxIT binding to calcineurin in vitro, which I'm not taking the time to show you today. But what we ultimately wanted to know was whether or not these interactions are occurring in vivo. And so to do that, we performed proximity ligation assays, or PLAs, on Hela cells with antibodies directed to either calcineurin or either of the 3 nuclear basket Nups. So you're using antibody pairs in these assays. Just to remind you, PLAs work by capturing proteins within about less than 40 nanometers of a physical distance. And they do this via rolling circle amplification off of species-specific antibodies between these protein pairs. So as shown, and that's schematized up here on the top right, sorry. So keep in mind that PLAs, at least the way that we performed them, are also able to capture the endogenous levels of proteins and also give you spatial information. So you're seeing where these interactions are occurring within the cells. And what's shown here on the bottom is that when we perform PLAs with calcineurin and either of the 3 nuclear basket Nups, we see robust PLA signals that really center on the nucleus, suggesting interactions between these proteins in vivo. Next, we wanted to determine whether these interactions were PxIxIT dependent, since we identified those PxIxIT motifs. To do this, we performed PLAs with the same antibody pairs, just on Hela cells that were expressing GFP peptide fusions, shown here above. So you either have GFP alone, GFP fused to a scramble peptide, or the high affinity PxIxIT peptide, VIVIT. And the expectation is if these are truly PxIxIT mediated interactions, expression of a competitive peptide like VIVIT might lower the PLA signal in those cells. And that's what I'm showing you here. And I'm just going to kind of draw your eyes to the result that we want you to appreciate, which is that in all cases with each of the nuclear basket Nups, when you express a GFP VIVIT peptide, you see a significant reduction in the number of PLA puncta per cell relative to GFP alone or GFP scramble. And this really supports the model that these are in fact PxIxIT mediated interactions that are occurring in vivo. But ultimately, though, we're interested in understanding whether calcineurin is dephosphorylating these nucleoporins. And I know I'm preaching to the choir here, you guys are a phosphatase group, but before we can really study dephosphorylation, we need to first identify any upstream kinases that might be phosphorylating these Nups. And so we went back to the literature and found that under very specific cellular conditions, a few of which I have listed here, that ERK, or the MAPK pathway, will phosphorylate various nucleoporins and disrupt nuclear transport, and that includes all three of the nuclear basket Nups. So this seems like a really good starting point for us. I'm going to highlight this little oxidant stress thing here because I'm going to come back to it later. So we wanted to test whether calcineurin opposes ERK phosphosites on these nucleoporins, which is exactly what we see. So what I'm showing you here are in vitro dephosphorylation assays on nucleoporins that were first phosphorylated with ERK. So what you're looking at is the amount of dephosphorylation over time when you introduce a constitutively active calcineurin. These experiments were really unique in that we also introduced in peptides during the dephosphorylation portion of the assay to see whether or not these dephosphorylation events were PxIxIT dependent and therefore more specific. So for Nup153 and TPR, what I hope you can appreciate is the lines with the squares on them. When you perform these dephosphorylation assays with that competitive VIVIT peptide present, you see a decrease in the rate and extent of dephosphorylation, really supporting the idea that these are PxIxIT-mediated specific dephosphorylation events by calcineurin. And again, more data that I don't have time to share today. Jaggery, who performed these assays, also did orthologous experiments on the budding yeast orthologs of all of these nucleoporins and found a conserved role for calcineurin in dephosphorylating these in a PxIxIT-dependent way, which is really exciting. Finally, we wanted to go back into cells and see whether or not calcineurin is regulating the phosphostate of these nucleoporins in vivo. So to do this, we went to Hela cells, which were treated in the same conditions as that transport assay was done, where we saw an effect on transport. So we want to see, under those same conditions, is the phosphostate of those proteins affected? So we performed that in Hela cells and then used this antibody called MAB414, which recognizes multiple nucleoporins, which I'm showing you here on the right. We can immunoprecipitate them and then perform mass spec analysis to see differences in phosphorylation between the treatment groups. And so what I'm showing you here is that down below in these little lollipop diagrams, is that multiple phospho-sites on these nucleoporins were calcineurin dependent. So any gray circle is an unchanged phosphosite, and then the red circles with either no outline or an outline were identified as being significantly increased in phosphorylation in the calcineurin inhibitor treatment in either 2 or 3 out of 3 replicates. I'll point out just as one example that this serine 529 phosphosite on Nup153, which we know is an ERK phosphosite, was identified as being calcineurin dependent. And this is really exciting because another group has shown that when this site is phosphorylated on Nup153, it interrupts the interaction with transport machinery. So it really suggests that this could be part of this role for calcineurin at the nuclear pore. Okay, so in conclusion, I've shown you that our SLIM-directed multi-pronged approach has identified hundreds of novel candidate calcineurin substrates and interactors. We've uncovered completely novel areas of calcineurin biology as evidenced here by our work on nuclear transport. In the future, we're interested in understanding more mechanistically how calcineurin is regulating transport. For instance, how do calcium levels factor into these events? Also, is calcineurin kind of broadly regulating transport, or is it specific pathways or even mRNA export and quality control that might be affected? There's a lot of open questions that are really exciting. But my last little piece of preliminary data, thinking about context for these events, is when I mentioned earlier that ERK phosphorylates nuclear porins under specific conditions. One of them is during oxidant stress. And so when we treat Hela cells with the oxidant stress diethyl malleate, or DEM, we see a phosphorylation of various nucleoporins that are hyperphosphorylated when you perform the same treatment, but with an inhibition of calcineurin layered on top of it, suggesting that typically calcineurin is opposing those phospho-sites on these key transport Nups. So we're starting to kind of dig into ideas like this to figure out the context in which calcineurin might be acting. And with that, I would like to thank the Cyert lab, especially Jaggery, who was my my partner through this whole project, all of our collaborators, which really helped breathe life into this story. And then I want to point out that the Cyert lab is actively recruiting and hiring postdocs. So if you're interested in working on this cool project or something else on calcineurin, we would love to have you, and you can reach out to Martha directly. That's all I have.
Alrighty, thank you, Callie.
Caroline, I feel like we need to have, like, I don't know, applause recording or something at the end of these.
I know, it's so sad. But if anybody has like questions that they want to start off with, there's also a couple in the chat, so we can go with.
Cool.
Okay, I'm gonna start with the chat. I had some that came to me privately. One person asked, do you have proteins that are binding both to PxIxIT and LXVP sites or sequences?
Yes, we do have examples of proteins that we have identified. I'll say that we've identified both the PxIxIT and the LXVP. What's kind of interesting with that whole angle is that we think that likely all calcineurin substrates probably have a PxIxIT and an LXVP, but the way that we've gone about approaching this is looking for like kind of the strongest or best versions of these motifs. And it may be that a protein with like a really great PxIxIT may only need an okay LXVP. So we haven't figured out how to identify the kind of, you know, not lower confidence, but less consensus LXVPs that could still be functional, if that makes sense. But there are examples of proteins that both have been identified, like the NFAT proteins.
And then the second question was, do you see an effect on nuclear envelope structure or reassembly after mitosis?
So we have not looked at reassembly after mitosis. I will say that from all of my reading, the primary phosphatase that's involved with all the dephosphorylation events at the nuclear envelope at the end of mitosis is PP1. And it's been pretty clearly shown that it's the main factor. So we could certainly look at that, but we don't really anticipate there being a hugely obvious role for calcineurin there. From just my IF experiments looking at cells with calcineurin inhibitors, I have not seen any issues with envelope structure. Nothing that kind of jumps out, but we're kind of starting to look at those things now.
I have a question.
Yes.
One, that was a fabulous talk. I really enjoyed that talk. You did very well. So I don't know if you guys have seen, I don't think Rosie is here today, but out of the Sears lab, they have a really nice study that came out a couple years ago now, where nuclear pores are actually enriched in cancer cells. The actual number of nuclear pores goes up in cancer cells, and that the site of the nuclear pore isn't just for import-export, but rather also has euchromatin associated with it.
Oh, yeah.
And so they have shown that MYC actually binds to the the TPR and to Nup 153, and that this is an ERK-dependent reaction, and that it seems to be early response genes. So they took cells that went into like serum, and then as they come out of that kind of stressor, MYC basically goes to the nuclear pore, initiates these like quick response genes, and then migrates from the nuclear pore further into the nucleus to regulate all of these other genes. So it also seems to be kind of like a separation in the transcription machinery. And so I think maybe looking at that paper, I think they did some, you know, like ChIP-seq and things like that, and seeing if any of the genes that are associated with that are deregulated when you express your calcineurin mutants would be super interesting.
Yes.
Or even just regulation of MYC at that spot. I mean, I would assume that PP2A-B56 alpha is also maybe involved in that process, but it would be interesting to know if calcineurin is as well.
Yeah, that's something I didn't really have time to expand on that today, but yeah, a lot of nucleoporins kind of moonlight and have other functions, not just play a role in transport. We're really excited specifically about how clear of a result we saw with the TPR dephosphorylation and how PxIT dependent it was, because TPR is not only important for transport, but it also has a huge role in RNA export and quality control, but also, as you mentioned, like it's a scaffold for ERK phosphorylation there, and then also helps to regulate transcriptional events. So there's a lot to dig in specifically on TPR, and that's on the docket for sure.
Okay, and I have one more from the chat, and then I think we'll go on to the next one unless anybody has something pressing. It's kind of long, so bear with me.
No worries.
Do you have data on primary cells, and do you think the extent of the effect on nuclear transport could vary among cell types? Wouldn't a generalized effect of nuclear transport not be consistent with the limited side effect profile of calcineurin inhibitors in vivo?
That's a great point. The short answer is that no, we have not done any experiments in primary cells. I think that would be really exciting to be able to do. Currently, we don't grow primary cells in our lab. Would love to look into that. I think it could vary among cell types. My kind of pet hypothesis that I, would love to flesh out is that I think that there may be either a specific transport pathway or a subset of cargo that are more differentially affected by calcineurin inhibition, just because I agree, if you saw, if calcineurin inhibition kind of globally shut down transport, then I don't think we would have immunosuppressant patients walking around talking typically, So I do think that there's something specific happening, whether it's cell type specific or cargo or transport pathway specific is really an open and exciting question to answer for sure.
All right, we got anybody else? Okay.
There was one more. I have not seen any effect on nuclear export, but we also haven't directly looked at that quite yet because we need a kind of different type of reporter. So to be determined.
All righty. Well, thank you, Callie, for a great talk and taking the time to talk to all of us. And so I guess we'll move on to our next presenter, which is Iain Hay, who's a PhD student, and I highly commend being our first PhD student to present, because I know when it comes to me, I'm going to be terrified. But Iain is at University of Cambridge, and he's going to be talking about PTPRU as a pseudophosphatase.
Helps if I unmute myself. Okay, so everyone can see that, yeah, as well. Oh, so hi, yeah, I'm Iain. I'm a PhD student in Haley Sharpe's lab at the Babraham Institute, but I also do a lot of my work with Janet Dean at the Institute for Medical Research, both here at the University of Cambridge in the UK. So our lab is generally interested in tyrosine phosphatases and the biological processes that they control. And one example of these could be epithelial to mesenchymal transitions, or EMT. So EMT is essentially a dynamic spectrum of cell states, which can range between full epithelial to full mesenchymal cell types. And so epithelial cells have very stable epithelial junctional complexes, and along with showing apical to basal cell polarity, And then essentially during EMT, these cell junctions start to weaken and break down with associated morphological changes, resulting in increase in cell motility and often more invasive cell phenotypes. But what's also important is that this isn't simply a modular switch between two different cell phenotypes, but you get a number of hybrid or kind of intermediate states between both epithelial and mesenchymal. And so this process is important in a number of physiological functions such as wound healing, but obviously this all has to be really tightly regulated by cell signaling, as dysregulation of EMT is often observed in pathologies such as cancer or fibrosis. So what does this mean for phosphatases? Well, for us, we are very interested in receptor type tyrosine phosphatases. So this is the one subfamily that we are interested in. It has 4 protein members. They're all protein tyrosine phosphatase receptor types, so PTPRK, UM, and T. And they're grouped based on their common domain structure. So they have a large extracellular domain, and they're all thought to be involved in cell-cell adhesion. So we can see that they are located at the sites of cell-cell contact. I actually know from a previous work that this homophilic, this extracellular domain forms trans cell homodimers across essentially the 2 cell membranes of opposing cells. So they're located to the sites of cell-cell contact, and we know that this homodimer interaction is actually needed to stabilize their expression at the cell surface, as if you can take PTPRK knockout cells and culture them with wild-type cells, and you'll never see any of these receptors at the boundary between the two different cell types. And simply also in adhesion, if you simply take the extracellular domain along with the transmembrane domain and transfect them into suspension cells, that alone is sufficient to cause this really dramatic clustering phenotype in these cells. So kind of importantly, a few of these members of this protein family have been suggested to be tumor suppressive, and indeed we've previously identified substrates for PTPRK, and we found that these were all these substrates were proteins involved in junctional organization. And when we looked at PTPRK knockout cells, we actually saw that they're starting to undergo some of these kind of EMT-like hybrid changes, where we saw essentially morphological changes where the cells have lost a lot of height. And also when you look at the junctions, they're a lot less tightly regulated. They're kind of leaky and a lot less well organized. And also and more recently, some more data has come out to kind of agrees with our previous findings, where they found that essentially PTPRK may act to actively inhibit EMT. But I want to talk about PTPRU today, and this is because actually in this family, it seems to actually be kind of the odd one out. It's very different. So actually at the start of my PhD, there was actually no data on whether PTPRU was actually an active tyrosine phosphatase. And as I said, these members have been suggested to be tumor suppressive, but actually there was some previous data that suggests PTPRU may be oncogenic. But despite this kind of difference, if you look in interactomes with what these receptors interact with, there's actually kind of common substrates and interactors between PTPRK and PTPRU. And it also, when you look at the catalytic motifs and the phosphatase domains, you can actually see that these are very different. And one thing, actually, really important thing I forgot to mention right at the start is that, so this extracellular domain interaction is linked to intracellular signaling by actually tandem tyrosine phosphatase domains, where the D1 phosphatase domain is often thought to be an active tyrosine phosphatase, and then often the second phosphatase domain, the D2 domain, is actually an inactive pseudophosphatase. So the catalytic mechanism of tyrosine phosphatase is actually very well characterized. We know it's down to these 4 key highly conserved amino acid motifs. So the most important of these would be the PTP loop, as this contains the key cysteine nucleophile, which sits at the bottom of the active site. And then you have the WPD loop, which contains an aspartate, which acts as a general acid for catalysis. And then finally, the Q loop acts to, the glutamine acts to position a key catalytic water molecule. So the fourth of these is the phosphotyrosine recognition loop. This doesn't actually play an active role in catalysis, but is actually thought to determine substrate specificity by creating a pocket of sufficient depth as to exclude smaller residues such as phosphoserine or threonine. But I was really interested in PTPRU, as if you look in these highly conserved motifs, you actually see that it's actually quite divergent. So you have a glutamate in the WPD loop instead of an aspartate. So you keep this acidic nature of the residue, but it's not clear how this may impact its activity. And then in this loop, which governs substrate specificity, is actually very divergent, and it's the most divergent across all of the PTPs. So we thought there might be a case where PTPRU could be active or inactive, or it may actually have a different substrate from phosphotyrosine. So my first job back when I started my PhD was to purify a recombinant PTPRU D1 domain. So in these receptors, this is the domain that is usually the active tyrosine phosphatase domain. So really what became immediately clear was that actually we couldn't detect any activity with the PTPRU D1 domain. So using the substrate PNPP, which is essentially almost a non-proteinaceous phosphotyrosine that's great to use for in vitro assays. Simply when it gets dephosphorylated, it turns yellow and you can just measure the absorbance. But what we see when we're using PTPRU D1 domain is we have no activity. So we can take the D1 domain of PTPRK, which we know is active, and even at low nanomolar concentrations, you can get nice measurable activity. And in comparison with PTPRU, actually at very high concentrations or at very long time points, you're seeing no significant activity over the baseline. And so this is a kind of in vitro microplate assay. So to make sure there wasn't any kind of cellular cofactors necessary for the activity of PTPRU in cells, we do dephosphorylation assays in cell lysates. So to do this, we took MCF10A cells and we treat them with pervanidate, which is a potent irreversible tyrosine phosphatase inhibitor to boost the levels of phosphotyrosine in our cell lysates. So then we take these lysates and then incubate them overnight with recombinant PTP domains and do Western blots to assess the total levels of phosphotyrosine. And again, essentially what we find is reflects what we saw in our microplate assays. So again, for PTPRKD1, the active domain, we see large-scale dephosphorylation of phosphotyrosine in the lysates. But again, for PTPRU, we can see no activity against phosphotyrosine. And it's actually similar to, so as I said, these D2 domains in these receptors are pseudophosphatase domains, so you see no activity. So at this stage, it seemed like PTPRU was similar to the pseudophosphatase domain of PTPRK, and it didn't seem like it actually could dephosphorylate phosphotyrosine. So one kind of caveat with all these experiments is what we were using the D1 domain in isolation, and there is some previous data which shows that having the presence of the D2 domain in your construct can actually influence the activity of the D1 domain. So then we wanted to essentially assess PTPRU's activity in the context of a full intracellular domain. Unfortunately, we weren't able to make this in E. coli like the D1 domain, but we were able to successfully express it in 293 T cells and also mammalian cells. So then simply did immunoprecipitations of these receptors from the mammalian cells, and then using PNPP, check their phosphatase activity. And really what we saw again was the same result. So again, with PTPRK, the active receptor, we see nice measurable activity when we IP it from cells. And we can compare this to essentially if you mutate out the catalytic cysteine to a serine, you lose all the activity. So this inactive mutant shows the exact same activity profile as PTPRU. And so again, it looks like PTPRU was indeed inactive against these substrates. Yeah, so this is just a nice bonus piece of data. So after the last time point, I actually left these IPs with the reaction mix overnight on the bench, and this is essentially what you see. So this is like 24 hours or something, and still you see no dephosphorylation by PTPRU. So as I mentioned, this loop that governs substrate specificity is actually very divergent in PTPRU. So we wanted to test some other phosphorylated substrates apart from simply phosphotyrosine. And so one of these we checked was if phosphatase species. So we sent these to Maya Cohen and at the University of Freiburg, who kindly agreed to run some PET assays for us. But again, we saw no activity for PTPRU against then their positive control. And so really, just to try and throw the kitchen sink at this, the last roll of the dice, we then checked substrates which are linked through a phosphoramidate bond. rather than the usual phosphoester bond that you're probably used to seeing. So this is essentially just phosphorylation linked through a nitrogen rather than an oxygen. But again, we couldn't detect any activity for PTPRU against any substrate that we tried. So in the absence of an identifiable substrate, we next set about trying to determine the mechanism by which PTPRU had became inactive. And as I mentioned, it has this key substitution in one of its catalytic residues. So we want to check whether this could solely account for its inactivity. But also we can't investigate this in the context of PTPRU as we can't detect any activity. So instead, we first assess the enzyme kinetics of the active PTPRKD1, and then essentially backmutated in this substitution. So we put the WPE into PTPRK. And what we see is that actually it does drastically reduce its enzymatic abilities. So it's about 100-fold slower in its turnover, but it does actually still have measurable activity, which we can't see for PTPRU. You can't do these assays with PTPRU because you just get no absorbance at all. And then also key, we can try the same thing. We can put the WPD back into PTPRU, but again, it does remain completely inactive. So it seems like this substitution couldn't solely account for PTPRU's inactivity. So to get a better look at how all these divergent motifs and things like that were impacting the catalytic mechanism, we then went to the atomic levels. So we put the pure protein down in crystal trays, which thankfully grew crystals. We took these to our local beamline in Oxford, and we were able to solve the crystal structure of the PTPRU D1 domain. So really one of the biggest changes we saw in the PTPRU D1 domain, so in this loop that governs substrate specificity, which is very divergent in PTPRU, in our structure, it was actually disordered, so we weren't actually able to resolve it. So this is the corresponding loop in PTPRK. But what we find if we look at, essentially here we've taken every PTP which has at least one cell structure and done a structure overlay, and you find that this loop is always ordered in these structures, and actually it has a very highly conserved secondary structure as it loops around here towards the active site, where this key tyrosine residue forms one wall of the active site. Yeah, so this is the kind of normal context that you see where this, the key tyrosine of this loop forms one large wall of the active site, creating this nice deep pocket for the phosphatase substrate to gain access to the cysteine nucleophile at the bottom of the active site. So the biggest other change we saw for PTPRU was in this PTP loop, which contains this key catalytic cysteine. And you can see again, this is the same large structural overlay of different PTPs. And again, it has a very highly conserved secondary structure in this loop. But again, PTPRU was completely different. So it has this large kink in the active site. And it seemed that actually this threonine residue in this loop sticks right into the middle of the active site. And when you look not just within this family, but across all the classical PTPs, this threonine is a unique residue at this position. So you never usually see it here. So we actually thought this kink conformation was unique. And it is in a way, with caveat that it is unique for a phosphatase in which the catalytic cysteine is reduced. So we actually found, so these cysteines don't undergo oxidation, and in the case of PTP1B, which is essentially the proteotypic tyrosine phosphatase, you can see from this video, so this is essentially a morph between the reduced and oxidized structures of PTP1B. So you can see when it becomes, this cysteine becomes oxidized, you get this change in conformation where actually this PTP loop starts to look like this large kink that you see in PTPRU. And again, this is the phosphatase recognition loop, which is disordered in the PTPRU structure, also undergoes conformational changes upon oxidation. And really importantly, oxidation of this cysteine will inactivate a PTP. So oxidized PTPs are inactive, so it almost seems that PTPRU is essentially evolved to look like an inactive tyrosine phosphatase. And so combining this structural information together, So as I said, this phosphatase recognition loop forms a large wall of the active site and forms these favorable interactions with the phosphatase substrate, allowing access to the catalytic cysteine. But because of these structural rearrangements in PTPRU, so you've lost your phosphatase loop, we're not entirely sure where it is. And then we have this rearrangement of the PTP loop in a way that essentially just seems to completely block off access to the catalytic cysteine, and it's really hard to see how substrate could gain access to the active site. So this could be the reason why PTPRU shows no tyrosine phosphatase activity. So next, with this information from the structure, we're then trying to set about to see which of these alterations actually could account for PTPRU inactivity. So to do this, we essentially made reciprocal chimeric proteins in which we've essentially swapped the phosphotyrosine recognition loops from PTPRK into PTPRU and the reciprocal version for use in our in-lysate dephosphorylation assays. So first, just looking at our PTPRK-based constructs. So again, the wild-type PTPRK causes large-scale dephosphorylation of phosphatyrosine. And actually, quite importantly, so this unique threonine in the PTPRU active site, if you put this into PTPRK, actually it's not sufficient to inactivate it. But what we do see is if you put this very divergent loop, which is disordered in our PTPRU structure, if you put this into PTPRK, this is actually sufficient to kill the tyrosine phosphatase activity of the enzyme. And then moving on to PTPRU, we see the same kind of trends. So the threonine wasn't sufficient to inactivate PTPRK, and we get the same thing if we remove it from PTPRU, that doesn't inactivate it. And we see the same thing regardless. So if you combine removal of threonine with restoration of the WPD loop, again, it remains inactive. And if you combine all of these mutations with putting in the canonical phosphatyrosine recognition loop from PTPRK, again, it still remains inactive. So it seems that it's a real combinatorial effect of all the different changes in PTPRU which account for its activity, and really no sole alteration could account for this. So by this stage, we are pretty convinced that PTPRU was a pseudophosphatase. So it has two phosphatase domains in its intracellular domain. But we did know from our previous reading, essentially, that PTPRU does have this overlapping substrate on binding partners with PTPRK, which is an active phosphatase. So one idea that we had was that, okay, can PTPRU bind these substrates first of all, and could it essentially act as a competitor for binding of these substrates? So in our previous work, we had identified substrates for PTPRK, the active phosphatase, and essentially what we were able to show was that the D2 domain, so this is the pseudophosphatase domain of these receptors, is actually key for specific binding of substrates. And we used chimeric proteins where we'd swap the different D2 domains of the family, and we'd interrogate their substrate binding profile. Essentially, so things like if you put the PTPRN D2 domain onto PTPRK, it can bind some but not all of PTPRK's substrates. So in a similar vein, to look at the binding of PTPRU substrates, I made a chimera intercellular domain which had the PTPRU D2 domain on it, and then we do recombinant protein pull downs from cell lysates to assess its ability to bind substrates of other phosphatases. So just looking at this previously identified PTPRK substrate afidin, which is a cell junction regulation protein. So we know that it binds PTPRK, and we were able to replicate that using our PTPRK constructs. And this is one of these ones which PTPRN cannot bind. So you get this very specific interaction. And it had been suggested in these previous interactions that PTPRU can bind afidin, and we were actually able to confirm that. So in our pull downs where we have the PTPRU D2 domain, we actually see this nice interaction with afidin, and just in comparison, this other substrate PTPRK is also a junction regulation protein, P120 catenin. All three of these constructs were actually able to bind this. And importantly, in subsequent experiments, we were able to show that recruitment of these substrates by the D2 domains was sufficient to allow dephosphorylation by the PTPRK D1 domain. But obviously in the context of PTPRU, where the D1 domain is inactive, you're not going to get this phosphorylation occurring. So again, previously we had been able to show that if you knock out PTPRK from epithelial cells, this leads to hyperphosphorylation of substrates. So we showed this by tyrosine phosphoproteomics, but you can also see it by Western blotting for specific phosphotyrosine sites. So again, looking at P120-catenin, we found this is our previous work where if you knock out PTPRK, you get hyperphosphorylation of these substrates, which essentially leads to junctional disassembly. So again, to look at the biology of PTPRU, we want to see essentially what the effect on this is in the context of a PTPRU knockout cell. So really what we saw is first we were able to replicate this result where in PTPRK knockout cells you see hyperphosphorylation of P120-catenin, which agrees with knocking out of the phosphatase which dephosphorylates it, but then actually surprisingly for PTPRU it can bind these proteins, and we found that when you knock out PTPRU you actually get a reduction in the amount of phosphorylation of these substrates versus what you see in wild type cells. So this essentially led us to propose this kind of hypothesis where PTPRU can bind these proteins and protein substrates of related but active phosphatases and essentially act to sequester them away from the active phosphatases and protect them from dephosphorylation. So essentially, when PTPRU is no longer there, you're freeing up more substrate for dephosphorylation by active phosphatases. And this is all published in NatComs 2 weeks ago today exactly. So if you want any more of the nitty-gritty, please go and read it. So that just leaves me to say thank you to Haley and Janet for their great supervision, and thank you to all of our collaborators for their help. And I'm happy to answer any of your questions.
That was great, Ian. That was a lot of work. That was very impressive. I actually have a question to start it off, and it might be in the manuscript that you talked about. Did you look at any of the other PTPRK substrates that you showed following PTPRU knockout? So basically, like that last slide, did you look at anything other than that P120?
We didn't, mostly due to the limitations, and for those experiments, we need to have site-specific, like we need antibody-specific for those specific phosphotyrosine sites. Obviously, we could look at it by something like, in the PTPRK paper, we did it by tyrosine phosphoproteomics. So we compared wild-type to PTPRK knockout cells. We could look at more substrates, interactors of PTPRU, by tyrosine phosphoproteomics, but it wasn't really something that was in the scope of that study really, because it's quite a bit of work.
Okay. Any other questions?
So have you guys seen PTPRU misexpressed in cancer cells?
So it is in kind of large data sets. We haven't looked specifically by our hands to see it. We haven't really looked at its expression in different cell types by Western blot or anything like that. But it is overexpressed in certain types of cancers. I think bladder cancer is the kind of top hit for that.
I have a general question about pseudophosphatases, if I may.
Yeah.
Does the profile, so if the model is that the pseudophosphatase function is sequestering substrates, right, protecting them, from dephosphorylation, what's known about like how the profile of pseudophosphatase is modulated? Does that change with EMT or cancerous versus non-cancerous or other different cellular states?
Good question. Not one I may have thought about before now. So
Yeah, and I realize that's kind of a big, broad kind of physiological question, but I've been interested in pseudophosphatases, for a bit of time, having seen some of them highly expressed in some of our favorite cells. So it's in the back of my mind.
I'm not entirely sure. I'm sorry. Maybe if Haley knows or not, but
I think equally it's interesting to look at the pseudophosphatases. They've not been massively studied. It certainly has an oncogene profile, so maybe understanding this conformation it adopts would be interesting in terms of interfering with its activity, and we'd really like to explore what it's doing in EMT and development.
Then we have one more in the chat. None of the PTPRU D1 mutants showed activity. What do you think is needed to generate an active PTPRU D1?
yeah, so kind of tried pretty hard to reactivate it. There is a lot of other, I haven't had a proper really good look. There's a great review on all the different motifs and PTPs, and there's some good structural ones. In our manuscript, we do talk about one kind of strange part of the structure, which is based on a unique cysteine. But I haven't done any mutational analysis on that one cysteine to see if it could. It just becomes very hard because you essentially are feel like I'm going to have to turn it into PTPRK before it becomes active again. We've had the same problem. We previously tried to make the D2 pseudophosphate made of PTPRK active, and we also failed to make that inactive again. But yeah, I'm not sure what it would take.
Mm-hmm. Okay, and I lied. I have one more, and it's two-part. Do you think that the active site is still important for interactions, so to protect possible substrates from dephosphorylation, given that it's occluded in the structure that you produced? And then two, do you expect that there are alternative conformations when the site is available for binding that are occluded in the conformation that you resolved being more of a regulatory feature?
Yes, so actually, the stuff about the active site being blocked, we're pretty sure that this isn't important for the interactions, as we think that these essentially what we saw in the chimera experiments is that it's the D2 domain that's actually very important. So essentially there would be a large binding interface between the D2 domain and the substrate that's being recruited for the receptor, and so then therefore the fact that the active site of the D1 domain is blocked, which wouldn't allow access to phosphotyrosine, but actually this isn't actually going to adversely affect the ability of the full modular protein to bind to the receptor. And actually, in the PTPRK paper, we do show that binding of a lot of these substrates is independent of their tyrosine phosphorylation status as well. Oh, and the second part, sorry. Yes, I guess the first answer also speaks to the second part, and so I'm not sure that there is any alternate conformations of the D1 active site. We did actually try to soak in substrates into our crystals to try and bring about a conformational change in the active site. But when we shot those crystals, we couldn't see anything bound. So we were trying really hard to try and bring about order to that disordered loop in the active site, but we couldn't see any conformational changes.
Mm-hmm. Alrighty. Well, thank you, Iain, and thank you, Callie. She had to jump off a little early, but I will send you guys a follow-up email with link if you want to send out the talks to anyone else, as well as a new sign-up sheet for September. Do you have anything, Jessie?
Yeah, the sign-up sheet will be September through December.
Mm-hmm.
Right.
Yeah.
Yep.
Alrighty. Well, then we will see you guys next month. Thanks for coming, everybody.
Yeah.
Thank you for organizing.
Yeah.
Really appreciate it.
Bye.
Bye, guys.
Okay.